So I'm Julia, I'm currently a 4th year medical student and I'm going to do a lecture on cancer today. So this is a operative room. If people could join it, that would be great. Let's leave it here for a second. And if anyone wants to put it in the chat to help people, that could be useful. We'll start off with some pathology for cancer. So some basic definitions. So hyperplasia is an increase in the number of cells, so when cell division occurs, and this can be either due to physiological or pathological conditions. But in hyperplasia, the cell growth will stop when the stimulus is removed, whilst in neoplasia, there's continuous cell growth, and so there's excessive autonomous cell growth, and this is caused by genetic mutations in neoplasia. So nomenclature, you don't really have to know that much about this, but this is just here for reference, I guess. So benign tumours tend to end in oma, whereas malignant tumours will be ending in sarcoma or carcinoma. It's just a useful tip to remember, although some cancers won't follow that rule. So as I said at the bottom, a hydratiform mole, which is a benign tumour of placenta, is a bit of a rogue one. But yeah, there are hundreds of different cancers, but these are grouped into 6 major categories. So you've got a carcinoma, which is epithelial origin, sarcoma, which is supportive and connective tissues, myeloma, which originates in plasma cells of the bone marrow, leukemia, which is bone marrow, lymphoma, which is the lymphatic system. And then you can also have mixed cancers, which are a mixture of these tissues. And this link at the bottom is just if you want any more detail on these cancers and their names. So the key difference between benign and malignant tumours is really that malignant tumours invade other tissues, whilst benign tumours don't, and also only malignant tumours can metastasize. So benign tumours tend to be slower growing, they tend to be well circumscribed and often have a capsule, whereas malignant tumours are invasive and usually don't have a capsule, and yes, there's also different amounts of differentiation. So cells from malignant tumours may not be differentiated at all, so they can be anaplastic, and they may be very pleomorphic, which means the cells all have variable size, shape, and staining. So this image at the bottom is of pleomorphic cells, so you can see they all have really different nuclei. So there's some big, some small, and they're all different shapes and sizes. And then benign tumours, although they're called benign, it's important to remember, they can have detrimental impacts. So they can lead to bleeding, pressure. In certain tissues, they can cause endocrine toxicity. So if it's a hormone-producing tissue. And there's always also the possibility that a benign tumour could progress to a malignant tumour. So tumours can spread in 2 ways. It can be local invasion or metastasis. So in local invasion, the tumour spreads directly into a neighbouring tissue. And whilst a benign tumour may be expansile, it won't penetrate into the adjacent tissue and is confined to that initiating tissue of the tumour. Malignant tumour can penetrate the basement membrane, as you can see here, and spread. And local invasion can compromise the function of involved tissues, either directly by destroying the tissue and preventing the normal tissue or organ function, or by compressing the tissue and the adjacent structures. Metastasis is a multi-stage process, and because metastatic cancer cells must be able to leave the tumour, survive in the circulation, and then survive and remain at another site in the body, and this is quite tricky. So microenvironmental changes at the site of the primary tumour can cause a local inflammatory environment, and this results in matrix metalloprotease secretion, which leads to the loss of normal organised tissue barriers. And then epithelial mesenchymal transition is a normal process in development, but in cancer, a change in expression of the factors involved in this transition in development cause a change from ordered epithelial cells to migratory fibroblast-like cells, enabling cells to break away from the tumour. And then the tumour cells can enter circulation, where they must survive before reaching the secondary site. But the secondary site isn't just anywhere in the body, so it enters a pre-metastatic niche. So the secondary site is the site where the circulating tumour cells could seed to. It has been suggested that exosomes, which are small circles of membrane that display specific integrins along with other molecules, are released from tumour cells and can direct the formation of a pre-metastatic niche in a specific tissue, with the integrin expressed by the exosome determining which tissue it will interact with. And cytokines secreted by the primary tumour can also promote the spread of cells into tissues. For some tumours, the most common sites of metastasis seem to be explained by the blood flow. So one study found that 66% of metastasis could be accounted for by blood flow patterns. This isn't always the case. So the seed and soil model suggests that the local microenvironment influences the chance of survival and growth of an arriving circulating tumour cell. So the microenvironment determines where the cancer cell is likely to seed to. But this theory still doesn't explain everything. So for example, why do contralateral organs, which would be the same tissue, rarely become metastatic site? And then to reach the site of metastasis, the circulating tumour cell must extravasate, so leave the blood vessel. And this often occurs via the formation of a microthrombus in the capillary, followed by retraction of the endothelial cells, so tumour cells can attach to the basement membrane. And then local proliferation will cause the basement membrane to rupture, so the tumour cells can enter the tissue. So malignant tumours can cause both local and systemic effects. Local effects are caused by things including the pressure that the tumour can place on surrounding tissues or organs, obstruction that the tumour may cause, hemorrhage if blood vessels are involved, and also infection. There's also intussusception of the gut, which is a condition in which part of the intestines slides into an adjacent part. And this can occur in young children, often due to unknown cause, but if it occurs in adults, it's normally due to a tumour. And systemic effects include cachexia, so wasting, hormonal effects, and also marrow destruction for leukemias and bleeding and anemia. An important association to know about is HPV and cervical cancer. So HPV is human papillomavirus, and it's an STI. It's a very common infection, and most people will get some type of HPV in their life, but the majority of HPV infections will not cause any symptoms at all. There are two groups of HPV: low risk, which mostly cause no disease at all, but can sometimes cause warts, and then high risk, which are associated with cancers. So the main association is with cervical cancer, also anal, vaginal and vulvar cancers, and also oropharyngeal cancers. Almost all cervical cancer is caused by HPV, and HPV is also thought to be linked to about 60 to 70% of oropharyngeal cancers. And in this high-risk category, there are about 14 high-risk HPVs, but of these, HPV 16 and HPV 18 are responsible for most of the cancers. Luckily, there is a HPV vaccine that is routinely offered to all children aged 12 to 13, and this can help protect against HPV infection and therefore against these types of cancer. So, Socrative question. I hope you all managed to log on. Which of these is a benign tumour? So I'd suggest looking at the last part to try and figure it out. Yep, everyone got that right. It's just adenoma, so the others, sarcoma and carcinoma, tend to indicate as malignant, whereas just the oma at the end is more likely to be benign. And the next question, which of these cancers is associated with HPV? Yeah, everyone got that right again. Just check you've been listening. Okay, that's great. Yeah, so cervical cancer is the main one people think of with HPV. Okay, so now moving on to, sorry, my slide seems to have, regulation of cell growth in tumours. So the hallmarks of cancer, these were initially described in 2010 and explain the 6 biological capabilities acquired during the multi-step development of tumours. So these 6 characteristics don't have to be acquired in a specific order. Random mutations will accumulate over time, leading to the development of these abilities in cancer. So the 6 hallmarks are, as can be seen on this, so sustaining proliferative signalling, evading growth suppressors. So the tumour must ignore that the surrounding cells are telling the cancer cell not to proliferate. Tissue evasion and metastasis, limitless replicative potential, inducing angiogenesis, so tumour cells still need a blood supply. An example from a mechanism for this is producing VEGF inducer, and then also resisting apoptosis or cell death. So to go into a bit more detail for a couple of these, so telomerase is involved in limitless replicative potential. So telomeres are structures that are normally present at the end of DNA strands that protect the chromosomal ends. This is in part needed because the mechanism of DNA polymerase prevents complete DNA replication to the end of the chromosome at every round of replication. So incomplete replication of the chromosome end occurs. And telomeres consist of non-coding DNA, so 6 nucleotide sequence repeats. And then in normal somatic cells, telomeres progressively shorten throughout life with each cell replication. And eventually the telomere loses the ability to protect the ends of the chromosomal DNA, and the cell stops dividing and goes into senescence. And telomerase is a reverse transcriptase that copies a short RNA template sequence within the enzyme into DNA, and it makes the shorthand and repeats found at the ends of the chromosomes of the telomeres. And telomerase is expressed in germline cells and early embryos, and also in some body cells. But in malignancy, telomerase, which is able to maintain the length of telomeres, is reactivated, enabling the cell to replicate continuously. So it prevents the cells going into senescence, so they can just keep dividing. And as well as extending telomere length, telomerase also has function in repairing DNA damage, which again helps the cell to replicate continuously. It's important to note that not all cancer cells do express telomerase. Some maintain telomere length through a different mechanism involving genetic recombination. So another hallmark of cancer is sustaining proliferative signaling. So a way of doing this is independence from growth factors. So normal tissues control the synthesis and release of signals that promote growth and regulate the cell cycle. And cancer cells deregulate these signals, meaning they can sustain chronic proliferation. Many growth factors bind to cell receptors with tyrosine kinase domains, which emit signals via intracellular signaling pathways and cascades. And so cancer cells can sustain proliferative signaling in a few different ways. So they may produce growth factor ligands themselves, which they can respond to via expression of specific receptors. So this results in autocrine proliferative stimulation. The cancer cells may send signals to stimulate normal cells within the microenvironment, and then these cells can supply the cancer cell with growth factors. So that can be in this diagram here. So, and the cancer cell can increase levels of receptor protein present on the cell surface, so the cell will be hyper-responsive to the growth factor. Mutations in receptor molecules can facilitate ligand-independent firing. So the ligand's not needed, so the receptor can fire by itself. And then the cancer cell can also constitutively activate components of signaling pathways that are downstream of the receptors, so no binding of the receptor is needed at all. So, for example, 40% of melanomas contain activating mutations in BRAF, which results in constitutive signaling through a mitogen activated protein kinase pathway. And another example is mutations in phosphoinositide, I don't know if I say, PI3 kinase catalytic subunit. So this is detected in different tumor types, but especially breast cancer. So the gain of function mutation is often found there. And this mutation hyperactivates the kinase. And PI3K generates PIP3 at the lipid membrane, which facilitates phosphorylation of AKT by another kinase enzyme. And AKT is the important one to remember. That's the central effector of the PI3 kinase pathway and transmits signals to many downstream substrates, orchestrating a variety of cellular functions, including growth, metabolism, proliferation, and survival. So they can lead to increased cell proliferation overall. And mutations or deletions that result in aberrant activation of the pathway are displayed as stars on this diagram, so the different things that could be mutated to affect the pathways. And opportunities for therapeutic intervention are also shown here, so the red lines, and that's just important as it demonstrates why understanding all these pathways and what's actually going on in the cancer cells is really important. Interestingly, these gain of function mutations in PI3 kinase are associated with a better clinical outcome, including survival. Anyway, in 2011, the same authors, so Hanahan and Weinberg, revisited the hallmarks and added 2 more. So deregulating cellular energetics. So the metabolic activity of cells is dysregulated in cancer cells, and also avoiding immune destruction. So the tumor cells must avoid being detected as being damaged cells by the immune system in order to be able to persist and survive and keep growing. They also added 2 enabling characteristics for cancer. So genome instability and mutation. So example, changes in repair mechanisms. So that's mutations in BRCA1 and BRCA2, the breast cancer genes. These two genes are involved in DNA repair. And also tumor promoting inflammation. So inflammatory cells secrete lots of growth factors that can help tumor growth. And these new hallmarks and enabling characteristics really emphasize the role that non-tumor stromal cells in the tumor microenvironment play. And just to demonstrate what I mean by the tumor microenvironment, these are some of the cells that would be involved. So bidirectional signaling occurs between tumor cells and non-tumor stromal cells, and also other cells in the tumor microenvironment. And some examples of the interactions and factors involved are given on this slide. So if you note in the center, there are sequestered growth factors in the extracellular matrix that the cancer can access. So an example of how they could access them is if the cancer releases proteases that would break down the extracellular matrix, thereby releasing the growth factors, enabling the tumor cells to grow. But there are lots of important interactions going on. Another important concept is cancer stem cells. So this concept proposes tumor growth is sustained by a subpopulation of cells that are highly malignant. And these cancer stem cells have been isolated from tumors and are the top of a hierarchy that's embedded in a genetically heterogeneous tumor. So that means within the tumor mass, not all the cells have the same mutations, and there'll be a number of different subclones within the cell and within the tumor, sorry. So this diagram sort of illustrates that. So some mutations may be shared by all tumor cells, whilst others are not. So driver mutations that occur early in tumor development are likely to be shared by all the cells, while mutations that occur later may only be present in a certain subset. So a couple more questions. So I don't think I moved on yet. So yeah, which of these hallmarks of cancer can telomerase play a role in? Yeah, excellent. Yeah, so it means cells can continue dividing. Next, what can a gain of function in PI3 kinase result in? Yep, okay, so I think most of you got this. So it's an increased activation of AKT. So that's the central molecule in the pathway I was talking about that orchestrates all the cellular functions. So there's a lot of molecules involved, but I think if you just remember PI3 kinase and hyperactivating mutation leads to phosphorylation of AKT, and then that can stimulate pathways that increase cell proliferation. So yeah. Okay. Then moving on to the genetic basis of malignancy. So mutations, so cancer results from mutations in DNA. So the development of most cancers requires a mutation of multiple genes in a single cell. And if you remember from the hallmarks of cancer we were talking about in the last section, for cancer to develop, multiple mutations occur in a stepwise fashion to confer the characteristics needed for the cancer cell to survive and proliferate. Increased rates of mutation or chromosomal instability can result due to mutations of genes encoding like DNA repair proteins, DNA polymerases delta and epsilon in the proofreading domains, and also cell cycle checkpoint proteins. And mutations in these genes have been found in most sporadic cancers. So I think dominant versus recessive mutations. So dominant mutations tend to get in proto-oncogenes, so that's a gain of function. And oncogenes result from the activation of proto-oncogenes, whereas tumor suppressor genes cause cancer when they're inactivated, and tumor suppressor genes tend to be recessive mutations. But I'll go into more detail of this in the following slides. So proto-oncogenes are normal genes that encode positive cell cycle regulators, so have a role in the regulation of cell growth, death, or differentiation. And when a proto-oncogene mutates or is overexpressed, it can become an oncogene and drive tumor development. So for example, a mutation in a cyclin-dependent kinase, which are protein kinases involved in regulating the cell cycle, could lead to the cell cycle completing and cell replication before all required conditions of the checkpoint are met. And this then means the cell cycle is dysregulated, which can lead to the development of cancer. So this is just A very quick reminder of the cell cycle. So it's really tightly controlled in most cells by multiple checkpoints. So you've got the first growth phase, goes into synthesis, then G2, then mitosis and cell division. And I've just put the R point there because that will become important in later slides. So that's one of the key checkpoints involved. So, viral oncogenes. Oncogenes were actually first discovered in viruses before they were discovered in humans, but viral oncogenes are developed from normal host genes that have been incorporated into the viral genome when the virus infected host cell before, and then they undergo mutation in the viral genome, which causes the normal host gene to become an oncogene within the virus. And then when the virus infects a host cell, it can transform the cell, usually via the integration of the viral gene into the host. And the viral oncogene can then confer properties characteristic of neoplasia on the infected cell. An example is RSV, so Rouse sarcoma virus. RSV here is not respiratory syncytial virus, which is confusingly another virus called RSV you may come across. But this RSV causes sarcoma in chickens, and it was the first oncovirus to be described. So as a retrovirus, it transcribes its genome, which is positive sense RNA, into complementary DNA before the genome is integrated into host DNA. And RSV was discovered by Peyton Roos in 1911, when the cell-free extract from a chicken tumour was injected into healthy chickens, which subsequently developed sarcoma, which is the tumour of connective tissue. And he was awarded the Nobel Prize in 1966 for this discovery. So it was later discovered that the SRC gene was responsible for the changes that occurred in the healthy cells leading to cancer, and SRC encodes a tyrosine kinase and triggers uncontrolled growth in the host cells. And you can see sort of the process of the integration and the viral replication cycle here. So RSV actually encodes the capsid proteins, which is GAG gene, reverse transcriptase, and the envelope gene itself. But this isn't the case for all oncogenic viruses. Some require a helper virus to provide one or more of these genes. But the oncogenic virus provides a selective advantage, as an oncogenic virus drives host cell proliferation, which therefore drives viral replication, so more viral progeny will be produced, which is advantageous for the virus. Yeah, so this is oncogenic virus that infects chickens, but since then there have been oncogenic human viruses discovered, such as Epstein-Barr virus. So some important virus and cancer associations for you to know are the ones in this table. So some viruses are associated or increase the risk of specific tumours. So human herpes virus 8, HHV8, is found in nearly all tumours in patients with Kaposi's sarcoma. Epstein-Barr virus, EBV, is a type of herpes virus, the virus is best known for causing mono or kissing disease, and it can be passed from person to person by coughing, sneezing, sharing cutlery or cups. And EBV causes a lifelong infection, even though symptoms disappear for most people after the first couple of weeks. And the seroprevalence of EBV in adults is thought to be about 95% worldwide. So it infects like a lot of people, but fortunately it only develops into cancer in a very small minority of people. So it can increase people's risk of nasopharyngeal cancer and also Burkitt's lymphoma, as well as some other cancers. But EBV-related cancers are more common in Africa and Southeast Asia. Both hepatitis B virus and hepatitis C can cause chronic viral hepatitis and increase the chance that someone will develop hepatocellular carcinoma, so liver cancer. HCV, so hep C, is more likely to cause chronic infection and lead to liver cancer than hep B, as only a very small amount of people go on to have a chronic hep B infection. Most adults will completely recover. A chronic infection is more likely if children are infected with hep B, whereas hep C mostly leads to a chronic infection with no acute symptoms. Just a note on HIV, which isn't thought to directly cause cancer, but does increase the risk of certain cancers, especially those linked with other viruses, such as Kaposi's sarcoma and cervical carcinoma, which is linked with HPV. This could be because HIV causes immunodeficiency, which might let other viruses thrive, Also, a weaker immune system may not attack the cancer cells, enabling the cancer cells to grow into a tumour without interference from the immune system. Luckily for these viruses, we have a vaccine for HPV and for Hep B. And there is now also a treatment for Hep C, which has a cure rate of 90 to 95%. So this has been a big development in the last few years. And this treatment for Hep C should stop it infecting people for so long, so will hopefully reduce the risk of cancer. So just a couple of questions on the viruses and their associations. So which virus increases the risk of hepatocellular carcinoma? Great, so yeah, Hep C causes a chronic liver infection, which leads to the liver cancer. And then, which is associated with Burkitt's lymphoma? Okay, great, yep, so most of you got this. EBV, so Epstein-Barr virus, and it's also associated with nasopharyngeal carcinoma. Then the other ones are listed there. Okay, so now on to tumour suppressor genes. Tumour suppressor genes normally prevent tumour growth when they're not mutated, and they can be mutated or lost in malignant tumours. And if they're mutated, they lead to dysregulated growth. So there are two main groups of tumour suppressor genes. Promoters, so mutation of these gets rid of brakes on cell proliferation and leads to cell transformation. And then the other group is caretakers, and these are genes involved in processes that maintain the integrity of DNA. And cells with mutated caretaker genes have a mutator phenotype, and as having a mutation in one of these caretaker genes affects the ability of the cell to repair non-lethal damage in other genes, that gives an increased chance of other mutations occurring that could lead to cancer. The evidence for tumour suppressor genes came from cell fusion studies initially, and these cell fusion studies enable you to create hybrid cells in culture that have the chromosomes of both parent cells, so you can then see which phenotype predominates. And it was found in experiments that fusion with normal cells suppressed the tumour forming capability of cancer cells, so the hybrid cells wouldn't form tumours. But if recultured multiple times, spontaneous loss of chromosomes occurs, and if normal chromosomes were lost, so the ones that weren't from the tumour cell, the ability to generate tumours would reappear, suggesting that there must be genes encoded on these chromosomes from the tumour cells. There must have been genes encoded on the chromosomes from the healthy cells that were suppressing the tumour. And so this is the recessive nature of the TSGs mutations. Some examples of loss of function mutations in cancer. So retinoblastoma, so a loss of function mutation in RB1 gene. Mutations in VHL lead to von Hippel-Lindau syndrome, which is characterized by large numbers of benign hemangioblastomas, which is a type of tumour. And then TP53 is mutated in lots of sporadic tumours, and there's also a germline mutation in Li-Fraumeni syndrome. And these loss of function mutations usually behave in a recessive manner at the cellular level. The P53 is an important molecule. It's often referred to as the guardian of the genome. P53, it blocks, P53 targets block CDK activity. So P53 normally turns off the cell cycle and stops cell proliferation, and it also mediates apoptosis. And P53 is activated in response to stress, so DNA damage, hypoxia, and also oncogene activation. So as oncogenes cause cellular stress, P53, if working normally, can eliminate many potential cancers before they even grow to a detectable size. So basically, P53 responds to cellular stress and turns off the cell cycle to stop the damaged cells from replicating. As shown in the diagram on the right-hand side, stable and active P53 activates transcription of MDM2, which is an oncogene that encodes a ubiquitin ligase, which targets P53 for degradation by ubiquitination. So this is a negative feedback loop. It means in unstressed cells, P53 is very unstable and will be degraded. And P53 is stabilized by blocking the MDM2 P53 interaction, which breaks the feedback loop. In cancer, P53 is sorry, I've lost my train of thought. So yeah, sorry, p53, I hadn't said already, is a tumor suppressor gene, and both copies need to be deleted for p53 to completely lose function and for the cancer to develop. So Lee-Fraumeni syndrome is quite a nice example of the two-hit hypothesis, which we'll discuss. The two-hit hypothesis, sorry, which we'll discuss in a minute. As in Lee-Fraumeni syndrome, there's a mutation in one allele of TP53, and then if the wild type allele, so the normal functioning one, is also mutated, then that leads to the development of tumours. A mutated p53 is found in over 50% of human tumour cells, revealing the important role it can play. And a loss of p53 function leads to a continued cell cycle progression without DNA repair and also a failure of apoptosis. So this means damaged cells don't die in a controlled way, but are instead able to carry on proliferating, which increases the risk of cancer. And there are a number of different mutations that could lead to a loss of p53 function, as well as a mutation in p53 itself. So for example, MDM2 overexpression would lead to increased degradation of p53, which could increase the risk of cancer. Loss of heterozygosity. This is a genetic event where a normal functional allele at a usually heterozygous location is lost. So at a locus in the genome where you've got one normal allele and one mutated allele, the loss of heterozygosity describes the genetic event where the functional allele is lost. So the locus of the genome becomes homozygous for loss of function. So you basically lose both working copies of the gene. And there are a number of different ways a loss of heterozygosity can occur, and some of these are shown in the diagram on the right. And this loss of heterozygosity plays a role in the two-hit hypothesis. So the two-hit hypothesis states most genes need two mutations to cause a phenotypic change. So like with Lee-Fraumeni syndrome, if an individual inherits a mutated allele in a recessive gene that gives an increased susceptibility to cancer, then unless a somatic loss of heterozygosity occurs, so the other allele mutates too, then cancer won't occur. And somatic loss of heterozygosity is rare on a cellular basis, but there's a high lifetime risk, as there are enough cells in a tissue to ensure that at least one cell will undergo an inactivating mutation. So cancer is almost certain to occur in the individual. And that's why, although it's a recessive gene, it can appear dominant. So an important tumor suppressor gene is RB, which is retinoblastoma. So the R point in the cell cycle is the point in a cell cycle at the G1 S checkpoint, and after this, the cell becomes committed to the cell cycle. So the phosphorylation of RB plays an important role at the R point. Non-phosphorylated RB binds to the transcription factor E2F, and if RB is phosphorylated by the CDK cyclin D complex, then the E2F is released and translocates to the nucleus, where it can lead to transcription of genes required for the S phase of the cell cycle, so the cell cycle can continue. R point deregulation causes increased risk of cancer, as then the cell cycle is uncontrolled, as the cell can enter the S phase without the normal cellular signals having led to this, and then the cells can proliferate uncontrollably. And at least one regulator of the cell cycle, such as cyclin D or CDK4 or RB, is mutated in most human cancers. This can be the RB protein itself could be mutated. So in the rare inherited condition of familial retinoblastoma, children inherit one mutated copy of RB, and then loss of heterozygosity occurs if the other mutates somatically, and this leads to cancer. So yeah, both copies of RB must be mutated. Increased degradation of CDK inhibitors can cause increased phosphorylation of RB, because then there's nothing inhibiting the cyclin CDK complex, and this can lead to cell cycle progression, or a mutation in the CDK so it is active without cyclin D, or overexpression of cyclin D can also increase phosphorylation of RB, which then leads to cell cycle progression and cancer. So, check the vague point got across there. If RB is phosphorylated, what's the effect on the cell cycle? Yeah, so this question is a bit of an oversimplification. But yeah, so the RB is important at the R checkpoint, which is between G1 and S. And if RB is phosphorylated, this means that the transcription factor is released and the cell cycle can progress past the R point into S phase. So our understanding of cancer. So the availability of a consensus human genome that we now have allows comparison with other genome sequences. And understanding the genes that can lead to development of cancer can help us develop new treatments. So for example, sequencing revealed that the BRAF V600E, so that's just a single nucleotide mutation, is present in about 60% of sporadic malignant melanomas. And this mutation leads to an alteration in the function of the BRAF protein kinase. And the structure of this protein kinase is such that selective inhibitors can be designed to target it. And this led to the development of vemurafenib, which is a BRAF inhibitor. And this drug induced a clinical response in 77% of metastatic melanoma patients with this specific mutation. And it was approved in 2012 in Europe. So it just shows how sequencing can help us find and target a specific mutation. The Cancer Genome Project uses high-throughput genome sequencing to identify somatically acquired mutations so that we can better understand and characterize the genes that are involved in cancer and the mutational processes and also the patterns of clonal evolution that are found in human tumours. And COSMIC is an online database of somatically acquired mutations found in human cancer. And this database is freely available to academic researchers. And if we are able to sequence the genomes of people who have cancer, we can identify specific treatments that will work well for people. It's giving us a chance to do personalized medicine. So for example, if there's an EGRF overexpression in colorectal cancer, cetuximab, which is a monoclonal antibody, is an effective treatment, but it wouldn't be if the tumor also has the KRAS mutation. So knowing specific mutations people have can really help us with effective treatments. Okay, so some more talkative, sorry, another viral one. So which virus is associated with Kaposi's sarcoma? Brilliant, yeah, you're supposed to remember. Yeah, HHV8. So the other ones, Hep C and B, a hepatocellular carcinoma, and EBV, Burkitt's lymphoma and nasopharyngeal carcinoma. And then, which germline mutation is Li-Fraumeni syndrome associated with? Brilliant, yep, TP53, so that encodes P53. So germline mutation in RB would be associated with familial retinoblastoma, and VHL would be von Hippel-Lars syndrome. Okay, so now on to cancer therapy. So just a quick note before starting, I just think this pie chart is important because we spend so long learning all the cytotoxic drugs that we often forget about surgery and radiotherapy, I feel, in second year. But yeah, surgery and radiotherapy are extremely important treatments and are actually the most effective curative therapies. So just to remind you to remember them. So moving on to chemo. The aim of chemotherapy can either be curative or palliative. So curative, you're aiming to kill all the tumour cells. You can have chemotherapy before surgery, which aims to reduce tumour size and treat micrometastases early, or it can be given post-surgery to treat micrometastases early and prevent them ever developing into secondary tumours. Or palliative chemotherapy aims to prolong survival and control the symptoms, even if there's no hope of curing and getting rid of the cancer. Cytotoxic drugs. The first type is antimetabolites. So a good example of these is methotrexate and 5-FU. So methotrexate is a folic acid analogue and therefore acts as a competitive inhibitor of dihydrofolate reductase. And it inhibits RNA synthesis and DNA replication. So stops the cell being able to divide. As well as being used to treat cancer, it can also be used to treat rheumatoid arthritis and also psoriasis and ectopic pregnancy. But side effects of it include myelosuppression, fatty changes in the liver, and mucositis, and it's teratogenic. So although it's really beneficial in cancer treatment, it's not great for its side effects. And 5-FU is a pyrimidine analogue. So it's bioactivated in cells to 5-F-DUNP, which covalently complexes to folic acid. And as these both target a similar pathway, so they are synergistic when used together. But side effects of 5FU again include myelosuppression, but this myelosuppression can be rescued with thymidine, and also another side effect is photosensitivity. So another type is alkylating agents. So alkylating agents in general attach alkyl groups to DNA, specifically to N7 of the guanine base of DNA, so G. Cyclophosphamide is an alkylating agent, and it's oxidized in vivo to form phosphoramide mustard, which is chemically similar to the mustard gas that's used in chemical warfare. So if you look at the structure, which is at the bottom of the slide, you can see there are 2 chlorine atoms, and displacement of these chlorine atoms allows covalent cross-linking between N7 nitrogens and guanine bases, and this causes cytotoxicity. The enzyme aldehyde dehydrogenase can detoxify one of the intermediates, so the drug has some selectivity for cells that have low ALDH. So for example, liver cells which have high levels of this enzyme tend not to be affected as much by the drug. And again, cyclophosphamide can also be used to treat some autoimmune diseases such as lupus and rheumatoid arthritis. And then cisplatin is another alkylating agent that also has 2 chlorine atoms. And so with cisplatin, water displaces the chloride ligand, enabling the cross-linking of DNA bases by platinum. And it's intra-strand DNA cross-links that are responsible for the majority of the cytotoxicity. And cisplatin is very effective in testicular cancer, although I don't think we know exactly why. So another class is topoisomerase inhibitors. So doxorubicin actually has quite a lot of effects. So it's an anthracycline antibiotic, but it also has anti-neoplastic activity. So it intercalates between base pairs in the DNA helix, preventing DNA replication. And it also inhibits topoisomerase 2, which stabilizes a cleavable enzyme DNA complex during DNA replication, preventing the ligation of the nucleotide strands again after the double-strand DNA breakage. And doxorubicin also forms oxygen-free radicals, which causes cytotoxicity. But this formation of free radicals is also responsible for some of the side effect toxicity of the drug. Microtubule poisons block mitosis. So vinca alkaloids, such as vincristine, inhibit tubulin polymerization, thereby preventing the assembly of the mitotic spindle, which causes the cell to arrest in metaphase. And vincristine can only be given intravenously as it's severely neurotoxic, so if it was given intrathecally, it would be lethal. And taxanes are also antimitotic drugs that prevent the disassembly of microtubules, and these are produced by yew trees. So adverse effects of cytotoxic drugs in general. Extravasation of cytotoxic drugs is a leakage of the drug into the local tissue at the site of infusion. This can be due to previous vascular damage, fragility of the vein, or just poor cannulation technique. And this extravasation underlies the inherent non-specific toxicity of the chemotherapeutic drugs. So what you really want is rapid dilution of the drug in the blood, rather than leaking into the local tissues. Reversible adverse effects include damage to the bone marrow, lymphoid tissue, GI epithelium, and hair. So bone marrow toxicity is a common cause of morbidity and mortality following cytotoxic chemotherapy. And a big one to look out for is a neutrophil count that's really low if bone marrow toxicity has occurred. So if you've got a low neutrophil count along with a fever, this is strongly predictive of bacteremia, so neutropenic sepsis. And this can be fatal, so it's really important that this is treated quickly with antibiotics. The reversible side effect is hand-foot syndrome, which usually occurs within days of starting chemo and affects the soles and palms of the hands and feet, which may swell, become numb, and also desquamation occurs, so you lose skin from the soles and palms of the feet. And these reversible ones, so this is in cells that replicate quite fast, so the cells will be replaced, I guess, after the drug has stopped being given. And then irreversible effects happens to cells that don't tend to be replaced and replicate as quickly in the human body, so damage to kidneys, nerves, heart, and lungs. And there's also therapy-induced tumorigenesis, which accounts for 1 in 6 of all new cancer diagnoses in the UK. Resistance can develop to chemotherapy, And a lot of these mechanisms are actually quite similar to resistance to antibiotics. So there can be decreased drug entry into the cells, altered expression or mutation of the target enzymes of the drug, improved DNA repair activity. So if the drug is trying to damage DNA, then yeah, DNA repair can be improved. Decreased tendency to apoptosis, so impairing the apoptotic pathways, improved detoxification of the cell, and also drug extrusion, like by using efflux pumps, for example. So it's important to avoid drug resistance. So to do this, you can use the maximum tolerated dose. So you're using as much of the drug as can be tolerated for a short period as possible. And then combination chemotherapy can also help, so targeting multiple aspects of the cancer cell biology simultaneously. And an example of this is CAV therapy for small cell lung cancer. So that is cyclophosphamide, adriamycin, which is another name for doxorubicin, and vincristine. So these all have different mechanisms of targeting and killing the cancer cell. So as you probably saw there, there's quite a lot of horrible side effects, and the of this chemotherapy, and the idea behind targeted cancer therapies is that they're targeting a specific pathway or cell rather than just causing DNA damage or inhibiting metabolism in general. So they're more specific and have lower toxicity. And targeted therapies, that can be hormonal therapies or molecularly targeted therapies. So starting with hormonal, tamoxifen is metabolized to hydroxytamoxifen inside cells, which is an estrogen receptor antagonist. So this binds to the estrogen receptor, recruiting co-repressor proteins, and then inhibits estrogen receptor dependent transcription. And so this helps prevent the cancer. And it may also have, it treats the cancer, and it may have additional activity against the cancer that's unrelated to this antagonistic effect too. But side effects include that it has the opposite action in the endometrium. But yeah, I think it's still a really good treatment. There's also aromatase inhibitors. So these stop the peripheral production of estrogen, but they can't stop the ovaries from making estrogen, which is why they're mainly used to treat postmenopausal women. But they're much more effective for treating postmenopausal women than tamoxifen is. And then testosterone reduction is a useful treatment in treating prostatic tumors. So reducing testosterone levels can help treat these, and this could be done by an orchidectomy, using drugs to depress LH release, so GnRH agonists, or by using anti-androgens. So anti-androgens can work by blocking the androgen receptor or inhibiting or suppressing androgen production. Imatinib is a molecularly targeted therapy, so it inhibits tyrosine kinase activity, and it's a drug that's really good for chronic myeloid leukemia. And this is because in chronic myeloid leukemia, there's a characteristic fusion protein, BCR-ABL, which is generated by the Philadelphia chromosome. So that's a translocation that's characteristic of CML. And this BCR-ABL fusion protein is a mutated tyrosine kinase that has deregulated tyrosine kinase activity that leads to a dysregulated cell cycle. And so inhibiting this mutated tyrosine kinase can stop the abnormal action and treat the cancer. And imatinib has a really high response rate in early phase CML with comparatively little non-specific toxicity, which is a big improvement compared to the other chemotherapy drugs we've been talking about. And this is an example of why knowing the genetic and molecular details of a specific cancer is important so that we can target it more specifically. Another example of a targeted therapy is trastuzumab. You may have heard of as Herceptin. So HER2 is a member of the epidermal growth factor receptor family of transmembrane receptors. And HER2 is overexpressed in about 25% of breast cancers. And Herceptin is a monoclonal antibody that's directed against the extracellular domain of HER2 and leads to downregulation of mitogenic signaling. And it may also induce antibody-dependent cellular cytotoxicity too, which would kill the cancer cells. And it can be combined with chemotherapy to increase survival and response rates further. But it's only effective in those cancers which are HER2 positive. Just a diagram to help you think about where all these different anti-cancer agents are targeting the cell cycle and inhibiting the cell cycle. So cancer is caused by a deregulated cell cycle and proliferation, and so the drugs will target different points. But as well as the therapies we talked about, there are lots of other therapies for cancer and more research to be done too to develop targeted treatments further. So this slide is talking about checkpoint inhibition therapy, which is a type of immunotherapy. And checkpoint inhibition therapy is actually already approved for certain types of cancer. So CTLA4 inhibitors, and CTLA4 is an inhibitory T cell receptor that's upregulated upon T cell activation. So yeah, CTLA4, it competes with CD28 to bind to CD80 or CD86 on antigen-presenting cells. But overall, it's just the CTLA4, if it binds to APCs rather than CD28, then it leads to decreased naive T cell activation and proliferation, so it leads to fewer T cells being activated. And so CTLA4 inhibitors promote T cell expansion, which means there are then more T cells that could go on to attack the cancer. An example of this is ipilimumab. And then another type of checkpoint inhibition therapy is PD-1 inhibitors. So PD-1 is broadly expressed and is thought to suppress T cell activity in the periphery. And PD-L1 is overexpressed on many tumour cells and is often described as acting like a shield protecting the tumour from the immune system. So the PD-L1 binds to PD-1 on T cells, leading to T cell anergy. So the T cells are inactivated, basically. And anti-PD-1 or anti-PD-L1 antibodies remove this protective shield from the tumour, so there are increased T cell responses against the tumour cells. So this is just one example of a different therapy for cancers, but there are other directions that could be taken too. So gene therapy, more personalised medicine, looking at mutations, targeting specific proteins, and yeah, lots of other fun advances too. So for time for a final couple of Socrative questions. So what's the mechanism of vincristine? Okay, yeah. So whether you got this right, so yes, it's a alkaloid, so it prevents polymerization of tubulin, stopping microtubules forming, so that stops the cell cycle in the mitosis phase. Then aromatase inhibitors. Great, yeah. Actually, you got that right. So yes, aromatase, they stop peripheral estrogen production. Aromatase inhibitors can't stop estrogen production in the ovaries, so that's why they're only useful in postmenopausal women. So when estrogen is no longer being produced by the ovaries, then aromatase inhibitors can be really effective. Thank you very much. That's the end. If any of you have any questions, please feel free to email me. And I'd be very grateful if you can fill in the feedback form, and you'll also receive a copy of the lecture handout if you fill it out. Thank you.